The computer here. All right. Anyway, thank you for coming to our capstone seminar. Already roll up. This is 25 years we've been here, so it's been pretty amazing run. So anyway, it's a pleasure having you here. We're gonna have this big bash, and it looks like I'm gonna have to buy more food if all of you show up. So we'll have plenty of food though. So it's gonna be a Park Place at 6:30. So please please feel free to come by from 6:30 to about 8:30 or more. We'll see how long the party until they kick us out. We'll have maybe a little podium for people to say some positive things.
I told him, I'm not really sure I want that to happen.
So anyway, he's done all sorts of amazing things. He's been here for 25 years. He's been the cornerstone for teaching genetics and parasitology and conservation. He's been a Fulbright scholar. He's got to go to Poland. He's taught over there. He's done research over there. I think I mentioned he also had a ENAS paper. He's just taught an incredible number of students. He's really made a legacy here, and he's been an amazing advocate for the farm. We really appreciate it.
Rich, before you get too far, what are we sharing right now?
I believe everybody should see the screen okay.
But I think we need a green, right?
Well, it's over here, I believe. Can everybody see the screen online? Can somebody chime in?
Yes.
Good screen.
Yep, you can see the actual presentation.
My slides? Okay,
Thank you.
good. Okay, I'll have this on for a second, just so I can say something. So thank you very much, everybody, for coming. It's been really fun for me to look back and think about things I've done, and so I'm really excited to do this. Some of you know that I'm a yacker. I'll be looking at the clock up there. When I was doing this, I was trying to decide if I should talk about my whole life. I decided no, and then I thought about all the research I've done at Western, and I thought no, I would have to explain too many different things. So what I'm going to do today is talk about some research that sort of goes in a circle. Like stuff I started with as a PhD student, drifted away from for a while, and I've come back to doing that. And it's all about looking at parasites in wild mice. So thanks, and so we'll start. So okay, so we start with the intro parasitology part. So oh. Yeah, thanks. I can never function without an editor. So thank you very much, Leslie. So I'll start with a super general background. When I say parasite, I'm talking about organisms that are symbiotic, meaning they live in close association for a long or short time, generally smaller than their host that they're getting energy from somehow. And in many definitions of the word parasite, we say that they are harmful to their host. But that question mark means that there are lots of things we call parasites for which we have never measured that. So we're just guessing, okay? Parasites include a wide variety of organisms, from multicellular eukaryotes to these weird prion protein things that are infectious. Organisms from this column can infect many of those, but people that call themselves parasitologists, like me and teach classes, generally focus on animals that infect animals. So animal parasites of animal hosts, sometimes protozoa. I will say all my talks that work today will be about animals infecting animals, although I have a student now starting to work on protozoa, and I'll maybe say his name later. Okay, so there are lots of parasites out there. Why do we care about them? From an ecological or evolutionary perspective, I will say to my class, well, it's likely true that parasites are the most diverse organisms on the planet. So think about, we are free living, most of us, not parasitic. And so all these free living hosts on the planet have one or multiple species of parasites living in them. So sort of by simple barroom napkin math, there are probably more parasitic species than free living species on the planet. Most of them are small, so they're actually really abundant, right? So they're everywhere. And another major reason we care about them is part of this definition of parasitism. They hurt things. So this is a table that's about 10 years old. Parasitology students, all this stuff will be on the final, right? But my point for everyone here is that we estimate that close to 1 million people are killed by parasitic infections every year. That's a small number of all the people that are infected. So a quarter of the people on the planet, largely in tropical areas, have some kind of parasitic thing in them, okay? Not only do parasites hurt humans, they also hurt the things we love, like dogs, cats, domesticated livestock. So we humans spend billions of dollars each year trying to prevent parasite infections or cure them, right? So they're a huge cost. Oh, in agriculture, right, they reduce crop gains and livestock production, so that costs money too. And not only humans and our special creatures, wildlife populations are likely affected by parasites too. So this is a picture of a moose with a heavy infestation of these ticks. As a result, their skin gets inflamed, they rub off their fur, they cannot thermoregulate in the winter, and they often die. So parasites can have important implications for individual health and population size in the wild. So let me make sure where we're going here. So since these parasites are everywhere and they have important consequences, a lot of my research questions have to do with if these parasites are out there and important, can we understand why certain hosts get infected and become more likely to be harmed by parasites in the world? So I did all my work on these mice called Peromyscus. I'll say more in a second. I just want to preview that, like, everything I'll talk about, and so this is why I chose these topics, because they're all kind of the same. Every project I do, I got and I kept some mice and I examined them for parasites on the outside or the inside. Then I measure other things like features of the host or features of the environment. And then the whole statistical analysis is to see if infection is correlated with other stuff that I measure, okay? And today I'm going to talk about 3 projects in the course of my sort of mouse career that look at these factors that could affect infection. So first is genetic diversity in host populations, then it's forest fires in the environment, and more recently, both host and environmental factors that could affect infections. I have been studying the genus Peromyscus, for 20 or 30 years. So this is a genus of rodents. They all kind of look like this, brown, but so, I've totally grown to love these. I think these are the most beautiful animals on the planet. So let me describe them this way. Beautiful cinnamon backs, lovely white bellies, the biggest puppy dog eyes you've seen on any animal on the planet, period. So beautiful. So they all kind of look like that, but there are lots and lots of species in North and South America. 2 of the most widespread and abundant ones are known as the deer mouse, Peromyscus maniculatus. And in this map, it's the distribution that's blue on the west, almost down to the southeast. Peromyscus leucopus is more southeastern. Down here in Florida, there are different Peromyscus species, but Peromyscus leucopus is also widespread and it goes way over in the west. There's a bunch of overlap between these 2 species and they can be hard to tell apart. And I'm not going to talk about that. But these are essentially, for a mammalogist, the model organism for mammals on the planet, right? So if you study ecology or evolution of mammals, most people do research on these mice because in almost every place you go, especially in North America, they're the most common and abundant mammal that there is there. Okay, so my first question is going to be this topic. The question is, does host genetic diversity affect the number of infected hosts in a mouse population? So that's me up in the picture, and this is my advisor, Phil Myers from the University of Michigan. And I put him up here because he essentially started my career. I showed up at Michigan, said, I'm interested in conservation. I want to study cheetahs or chimps. And he looked at me and shook his head and said, that's stupid. And that was not the first, Phil did not call me stupid. I interpreted it as being called stupid, but he basically said, if you study those big animals, tigers, cheetahs, blue whales, there are none of them on the planet. Your sample size is going to be so small, you will never actually be able to say anything. But I know an animal, and it's deer mice. And we're gonna say, so they're super abundant. There you can actually do science, measure things, identify patterns in nature. So, and he's the guy that turned me on to these places where I did my PhD work. So, this is a topic, this is just to show that, this is old stuff, right? Like about 2000. So I was publishing this after my PhD when I showed up here. Okay, so, I start with, the genetics intro. And so genetic diversity, so in this picture, the height of these sort of lines is population size of hosts. Time is this way. And there are these things called bottlenecks. And the idea is that in large populations, there's a lot of genetic diversity, okay? And most individuals are genetically different from each other. But if a population is reduced in size, this is the bottleneck part. It could be from overhunting or habitat loss or pick your favorite environmental disaster. But after a bottleneck, you end up with a population with reduced genetic diversity. Individuals are more like each other because diversity has been lost. Okay, so that's just population genetic stuff, right? The relationship this has to do with parasitism or parasitology is this. So there are some theoretical expectations about how you would expect disease to spread through genetically diverse and genetically uniform populations. So here's a cartoon that I probably showed, as a grad student, but the ovals are meant to be hosts. They are different colors to suggest they are genetically variable. That little space capsule there is a virus invading this population. And the idea in this picture is that the white color tells you that by some molecular mechanism, it's compatible with the white host, can infect them and hurt them. Okay, so in an outbred variable population, that virus will come in, infect a few hosts, but even if they die, the population will be left over because there are a lot of resistant individuals. In contrast, after bottlenecks, say, the population might be more genetically uniform. Most of the individuals are white here. The virus can attack all of them. If they die, you end up with a very small population that could go extinct. So this issue about whether genetic diversity protects populations from diseases is a really important conservation biology one, where people are concerned about small populations with low genetic diversity. When I was working on my PhD, I am really tangent prone, and I'm stopping me right now. I'm trying. Okay, at the time of my PhD, I got really into this idea, but I read the literature as hard as I could, but found no examples where people had actually gone into the wild and compared populations that had higher low genetic diversity and looked at parasitism. Like there was nothing. So this is the mouse I studied. So take a picture taken by my advisor. So this is a deer mouse. I studied them in northern Michigan. This is the worm that I studied. So these are two deer mice cut open, and you're looking at their livers. So this is a healthy liver, smooth and shiny and red. This is a mouse with an infected liver. And all of this white stuff is scar tissue, worm eggs, and dead and decaying worms, okay? And, I went up there and I was catching mice the first time. I'd never had parasitology class, but even I could see there was something in these mice, right? And so I was super lucky in that way. I stumbled onto these mouse populations that had these worms. Here's a slide from my PhD defense, and you're gonna see these in the next few minutes, but I kind of like them, right? You see all the hairs and scratches. I feel like this is one of those 1950s newsreels. And now, students, okay. Here is the host. Here are the worms in the body. And I think the only thing I want to say about this life cycle, I love life cycles, parasitology students know. Here's the egg. If you eat an egg, basically the egg hatches, the worm gets into the bloodstream, the babies like burrow into the liver where they mature, they start having sex and laying eggs through these those white tracts until they die. So an infected mouse has these worms in it that are stuck in the mouse. And that's really unusual. Most parasites come off the host, they're out of the host somehow. So this worm cannot get transmitted until this mouse gets eaten. Here is a coyote or a fox. eating and dissolving away the mouse, so the egg ends up in the environment, which could be eaten. What's more likely, we think, is that infected mice end up dying and other mice come along and cannibalize them, and that's how they get infected. Okay, so there are these mice, there are these worms. In my PhD, I went to 9 different sites in Michigan. 4 were on mainland sites, 5 were on island sites. And the reason I did this is, remember, I'm interested in genetic diversity or not. And it turns out that comparisons of mainlands and islands are often sort of natural experiments. In many cases, island populations have reduced genetic diversity because they've gone through a bottleneck getting to the island. So I did not know if they had low diversity, but I went there hoping they were. I caught tens of mice in each place. I measured parasitism as, my measure of parasitism was the prevalence of capillaria. So I counted the number of mice, counted the number of infected mice divided by the total, and prevalence is just the fraction of hosts in a population that have something. So 100% means they all have it, 0 means none have it. To measure genetic diversity, I had a slide of this, and it's like so old school. It's my last chance to show it, I think. I looked at something called allozyme diversity. Allozyme just means allelic versions of an enzyme. So I'm looking at protein diversity at 21 different genes. And I'll show you one picture so you have an idea of what that looks like. And so my real interest was only in, does genetic diversity affect parasitism? But I was going out in nature, and nature's complicated, right? And I was sure that not just genetic diversity would matter, but some other ecological stuff could too. One really important one is population density of hosts. And there's a lot of math that goes into this, but I will just say, can you imagine that in a given area, more hosts makes transmission easier if they're all crowded together? So with high population density, totally ignoring genetic diversity, you would expect more individuals to be infected. Okay. Oh, and what I'll say here is I measured population density by a simple way. It's called trap success, and it's just the number of mice I catch divided by the number of traps I set. So it's another fraction. So, right, set 100 traps, I can get 99 mice, which is 0.99 trap success, or I can get 2 mice, which is 0.02, and 0.99 is bigger than 0.02. And one tangent I'll say here for you grad students is, I spent about 3 weeks reading the literature on estimated population density. It's really super complicated. None of the estimates ever end up great. And so after 3 weeks or so of reading the super complicated math, I settled on this, which is just, I count the mice and divide by the traps. And that crude estimate of density is just as good as any of the other complicated ones. And I can do this in like my head. Okay, that's all. So yeah, this is a satellite picture of Michigan. University of Michigan is down here somewhere. But the point I want to make here is that I trap mice in island and mainland localities.
Well, island. Why would you trap mice on an island? No.
Oh, so mute yourself, please, Mark Benson. Okay. So I think you want to say distracted, but I just put this picture here. It says that this tangent is also, scratchy, but this is in my paper. I drew this by hand and rubbed these numbers on. That's all. Okay. Okay, there were some island sites too. Okay, and I just have to show these. So I had to get out of these islands. No one lives on these islands. So here's a guy who dropped me off in a dinghy. One time I got dropped off this way. Another time I sort of float plane that dropped me off somewhere. I guess I should say, I was almost always alone, but one time I had a camera crew.
No one here.
So these islands are beautiful. They are uninhabited, super, super scenic. I could go on and on. But now we get to the method. So I would set mouse traps. Here is a little orange flag telling me where the mouse trap is set. So I would set these transects out in the woods in these localities that were like a mile long, using a compass to make sure I didn't get lost out in the woods, which was not always successful. I did get lost in the woods, like overnight, a couple times, okay? So here's how I get trap success. There's me setting a trap. So it's a live trap where a mouse runs in. I set them out, I come back the next morning, and this is trap success. I count the number of traps with mice, here's the total, and so I get some estimate of density. Then I have to like see if the mice are parasitized or not. This is me cutting up in a mouse. Here's one of those livers. Some of you who have worked with me recognize all of these dishes with body parts, etc. So this is like 20 or 30 years ago. Here's a picture I found to say, my life has not changed one bit. I estimated genetic diversity in this population is this way. So this is an electrophoretic gel. Each spot here, each lane is a liver sample from a different mouse. And so you apply a current, these things move. Because of the quaternary structure of these proteins, you can tell the difference between homo and heterozygous individuals. This is a dimer composed of 2 subunits that move slowly, so SS. This is a different homozygote, and this thing is a heterozygote. So this is, so you can just basically count up the number of heterozygotes and use that as a measure of genetic diversity. Okay, so I want to get to these. So the statistics look like this. The outcome or the response was for each of these populations, the prevalence of that worm. The potential predictors were genetic diversity measured with allozymes and population density, misspelled trap success right there. And then the analysis I did, right, 20 years ago was just look for correlations between these numbers and that. The data were not normally distributed. I used some non-parametric statistics. But these are the results, right? So this is a graph in which I have enzyme heterozygosity here going up, the prevalence of infection here. Each dot is a different locality. And for years I would say, you know, those 9 dots took 2.5 years of my life. And that's what I got. But the main thing I'll say here is the correlation coefficient between heterozygosity and prevalence is negative, and it's significant, okay? Which is super cool, right? Like I said, I had never seen this before. I just read a review paper, a recent one, that this is one of the first time people ever actually looked. So I was super, super excited, right? It's like, oh, yeah, what people thought might happen did. And then I looked at population density. And so population density is here, prevalence is here. And do you see the pattern? It goes up. So where the mice are more densely crowded, a higher fraction are infected. It turns out that small islands have low heterozygosity and high density. And so it's like, oh shit, the 2 things that could matter might, and how do I tell which one matters? So we can do this. To ask that question, which of these 2 things is the important thing? We can look at partial correlation coefficients. And if we look at the correlation between heterozygosity and prevalence, holding density constant, or we're taking care of it, the relationship is still negative and significant. If we look at the relationship, the correlation between density, population density and prevalence, holding heterozygosity constant, it's still positive, but not significant. So when we compare the 2 things, it turns out in this case, genetic diversity is the major predictor. And to the best of my knowledge, this was one of the first papers to ever say this, partly because I just read that about me the other day. Okay. So here, this is a speed bar. So that was like prior to here. When I got to Western, I did some experimental mouse work, but I was sort of driven mostly by what students wanted to do. So I'll just say, I had a student, Kim, work on turkeys and their lice. I had a student, Krista, who worked on cockroaches and the pinworms in them in the greenhouse at the end of Wagner. And I had several students that worked on fish and flukes in them. So Beth started, Sammy and Shane continued this work. Maybe I'll say it in front of you. So we published Beth, Sammy and Shane's and our work still needs work, and I'm retiring. So that's what I hope to do. Okay. All right. So then, okay. About 10 years, I wasn't doing any field work with mice. Then I went on sabbatical and met this guy on the right. So this is Rafał Zwolak. He is Polish, but I met him in Montana, where he was a graduate student. But he is why I had then went on sabbatical to Poland a few years later. So the question I'll address here is, does fire in the environment affect average parasite load in mouse populations? And I guess the way I want to introduce this, I'll say why we might care in a minute, but like out west in Montana where he works, after forest fires, the numbers of Peromyscus explode through the roof. So after a fire, their population size increases, which is surprising, right? You torch the environment, you think it'd be bad for the organisms there, but these mice seem to do really well. He had, for his PhD, you know, the time when I met him and we were hanging out, he had studied things like predation in burned and unburned forests, food, foraging ability, et cetera, but found no serious differences between the two kinds of habitat. So, you know, we met and we'd go play pool and drink beer and do whatever. So 2 or 3 years after sabbatical, he said, hey, how about this? I've tried to rule out everything, but maybe parasites matter. You want to dissect some mice with me? And I said, oh, of course. It's like my one marketable skill is cutting up mice. Okay, so we work together. That's the paper. So for background here, I'll say, why would you expect any relationship between fire and parasitism? I'll try to explain that. So these are two, pixely versions of drawings I made for honor students. But on this left is a life cycle I drew of an endoparasite, a worm living in a mouse. On the right is an ectoparasite, a maggot called a botfly that lives in the skin of a mouse. But the main point I want to make is most parasites, although they're symbiotic and live in the host for a big chunk of their life, almost always have an external stage because they have to get dispersed. And so the whole idea here is that when they're parasites, if there's a fire, it might really hammer these external life cycle stages that then would not be available to infect hosts. Yeah, right. So the prediction would be that after a fire, there would be fewer parasites. So here I looked, so Raphael did the field work, I did some of the lab work, but here's a Peromyscus maniculatus, another deer mouse. Montana's out there. We did a survey of all the parasites on these mice. So that includes nematodes, roundworms, cestodes or tapeworms, so fleas, lice, and ticks. So some of these pictures are by a guy I'll show in a minute, Mike Kinsella. This is on my computer and I don't know where it is, what it is, where it's from. So, okay. All right, so we are going to be looking at mice for all these parasites to see if fire affects them. The work took place in like 2011. So there was a giant forest fire in Montana called the Davis Fire in 2010. In this study, we measure parasitism by counting every parasite on each mouse. So the last thing I talked about was what fraction are infected. Now we're actually keeping track of each individual and how many parasites it has. That's called abundance. And what we compare is abundance of different parasites that were collected either in burned or unburned plots around this fire. So transect. So there's a giant fire. So imagine this. I think I have a picture. You have a mountainside and half the thing has been fried. It's obliterated. I'll show you. But then adjacent to it is unburned forest that was the same before. And so we're comparing mice in those two places. You can see here that in the burned plots, we only needed 2 transects to catch a lot of mice. And in the unburned plots, we needed a lot of transects to catch fewer mice. So that's that population density thing. Okay, Montana's out west, that's all I'll say. Forest fires out west can be gigantic. Here's Raphael setting up trap lines a year after the fire. So I'll say this to all of you, so Shawn and people who worked at Kibbe. So the fire's out there, you know, this on the right is a picture 1 year after. It's a lunar landscape, there's like nothing at all. And that's in contrast, Dr. Jenkins has been doing burns out at Kibbe and it's not like that, okay. In this study, we trapped the mice. So Raphael trapped them, brought them back to lab. I started the dissections, and then Jonathan Vaughn was a master's student. He finished the dissections, he did all the identifying. Mike Kinsella is a retired parasitologist that happened to live in Missoula, Montana. And for years and years, he's been helping me identify things. So Jonathan did a lot of hard work, then sent stuff to Mike Kinsella to identify. And this is the stuff that Jonathan found. There's nothing for you to see here, except he worked really hard to be able to identify all of these different organisms. So you can see there's a whole bunch of different flea species, but only 2 are quite common. There's lice and ticks, a bunch of roundworms or nematodes, but only a couple are very common. And then there's some tapeworms down here. Okay, no tangents. Okay, so in this analysis that I'm going to show in one graph is basically this. So the outcomes that we're interested in is always some kind of infection estimate. I told you it's abundance. And here we're looking at the abundance of either ticks or lice or fleas or nematodes or cestodes. These are roundworms and tapeworms. There were too few infections by particular species to do it one at a time, so we just lumped these things together. Okay, potential predictors are fire. So that's the only thing I really care about here. Does having a fire where you live affect your infection level? But it's going to come up again later. The next part is like, we know that things like mouse sex or mouse size potentially can affect whether an individual is infected. But there are also other things like what transect we caught them on. Okay. So the analysis we did, and I should look around to see if my 501 chums are here. I see some. So statistically, we had to use an analysis called a generalized linear mixed model. So linear models just suggest that between your predictors or independent variables and dependent or response variables, there's some sort of linear relationship that you can model. And that's all I'll say. The G part here stands for generalized, meaning that your response variables do not need to be normal, or the error distributions after you fit some model are not normally distributed, okay? So with a generalized model, you can have any shape to your data. The mixed part means we have both main effects and what we call random effects. So the main effects are, we have replication, right? We have multiple sites that came from fires. We have multiple mice that are different sexes, but we don't have multiples of one individual. And so it could be that one mouse is more likely to get infected with everything relative to another mouse. And so these random effects let us deal with that kind of variation for which there's no replication, and we can just sort of tease out that. Okay. So these are the data. So we're going to have abundance or mean parasite load on this y-axis. The 4 different categories, 1, 2, 3, the 5 different categories. So 2 endoparasites that I have been calling cestode and nematode. So sorry about that. And 3 ectoparasites, tick, lice, fleas. These green bars show the average abundance of parasites for these different taxa. And I think the main thing you need to see here is like hardly any of the mice had nematodes or cestodes. There's like almost no green for you to see at all. Ectoparasites were much more common. And so then the big question is, when we put the bars up there for the burn plots, are the averages different? And this is the pattern, right? So no effect on the endoparasites. There were hardly any in either place. But when we look at the ectoparasites, no real difference in the lice, but a significant reduction in tick abundance and flea abundance. So in the burn plots, these mice on average have fewer ectoparasites on them. So lower tick and flea abundance after fire. The biggest effect here is on fleas. The potential important implications of this are, I think the first is, there are some data looking at tick numbers in the environment after fires. Actually, papers that we found after writing this paper and finding no references. So this sort of thing has been seen before, but no one has looked at fleas. Oh, this is a tangent I have to tell. So I started this work interested in a worm, right? An internal worm. I've always been a very worm-focused guy. And so this project started, so how this, like, yeah, I think I have time. This project started with me saying, okay, I don't trust the ectoparasite data. You know, you get them in a snap trap, the fleas are gonna jump away, or the ticks are gonna crawl away off of this cold dead mouse. I don't trust that the ectoparasites we count will accurately reflect what's on them in life. But the worms, I know they're not going anywhere, right? They're dead. It's gonna take days for them to crawl out the anus or the mouth, and we've got them before that. So we're gonna find the worms. Okay, so I wasn't even gonna look at ectoparasite stuff. It was like worms. I trust those data. So I dissect the first mouse. I put it in its bottles, whatever. I look at my dissecting tray, and there's a flea laying on it. And I think, oh my gosh, okay, there's fleas. Maybe I should look. And I did. And so it turns out that this is really important. I want to get to the last piece. So I think what I'll say here is one important implication of this. We don't know why this might be the case, right? The fire may burn up the mouse burrows with the fleas living in the nests. We don't know that. We haven't done any work. But, you know, it's possible that with fewer fleas, that allows mice to do better, right? They're sort of like better able to. This is like totally outside my area of expertise, but it's behavioral ecology stuff. There's research that suggests that mice that have fleas, they have a hard time foraging, right? Because they're all scratching. And so they're worried about predators. And so they can't really focus on the food because they're focused on scratching. But it could be that fewer fleas lead to more mice. And that has important implications for forest regeneration, actually, right? Because mice are omnivorous, they are seed predators. And if there are seeds in the environment, these mice are going to be all over them. So if their numbers go up after a fire, it makes it less likely that a forest will regenerate because of their presence. Okay, so my last one that I'm going to talk about here is work I'm doing now out at Kitty. So I'm going to say all that stuff. My principal collaborators here are Dr. McPherson and Dr. Paul. You see, I had to crop them differently to get their research creatures in. And so this question is what? So first I looked at sort of population, the host genetics. Then I looked at the environment and fire. This, I'm going to look at both host and environment factors that affect two different species of ticks that infect mice. This is a thesis by, how do I know her now? Ebby, right? First it was Stephanie, now it's Ebby. So Ebby, who just defended her thesis a week or so ago, and Dr. McPherson and Paul have been working with me a lot on philosophy and writing and statistics. Okay, so why are we doing this work? So I guess I do need to make this sort of snide comment. So I'm a worm guy. But these days, all my students is like all about vectors. Sada, are you out there somewhere? Kofi, so yeah, so okay, let me get back on track. So I'm a worm guy, but now everyone and their dog wants to study these ticks, etc. And so why do we study ticks on mice? And I need to thank Jasmine, so I'm using it a second time. So a student in my parasitology class took this picture of the ticks we're likely to find in Illinois. We care about ticks and their abundance in the environment because they can transmit a variety of diseases to humans. And I don't even think I'm going to name them, but deer ticks transmit Lyme disease, dog ticks and Lone Star ticks can transmit other stuff. Oh, should say something about Lone Star ticks, these ones on the bottom, they're the ones that cause alpha-gal syndrome or red meat allergy. Okay, so humans care about ticks because ticks are the major vectors in North America. At Kibbe, there are multiple tick species, and so we were motivated to understand infestation patterns by multiple tick species. And here's a general sort of epidemiological reason why. So co-infestation of ticks on mice allows for the possibility of this thing called co-feeding transmission, which I learned about 2 weeks ago, which is why I want to say it today. All right, so why would we care about how many ticks infest the mice and whether they are different species? Well, vectors like ticks get infected by sucking blood from infected hosts. So an infected vector has to inject a disease, and then other uninfected juveniles have to suck that up. So you need to have the pathogen in the host to get transmitted. The most common way that people think about this is called systemic transmission, where one vector inserts the pathogen, it then multiplies, and any parasite that comes after will suck that up. So that's called systemic because the infection is throughout the body. But this co-feeding transmission thing works like this. If you have an infected vector, it can be spilling out parasites into this reservoir host, the mouse, and any other hosts or any other parasites co-feeding at the same time can slurp up the parasites without the host actually becoming infected, right? So it's this crazy thing that I just learned about, but it makes it really important to understand how often do multiple tick species end up on one host. And so our question was going to be is, can we describe the patterns of multi-species tick co-infestations? I did this work at our field station on white-footed mouse, Peromyscus leucopus. Illinois is about there. The ticks, so I'll show you a map. WIU has a place called the Alice L. Kibbe Life Science Station west of here. Undergrads should take summer classes there. It'll change your life. Out at Kibbe, field work by Dr. Jenkins and some others tell us that these 3 species of ticks are there: deer, dog, lone star. Here are some ticks I just want to show you because this is cool and it shows you what we're doing with these mice. So in these bottles are different life cycle stages of the ticks. So the bottom 2 here are adults. So that's what you might see on you. In the middle, AAN is a nymph, and on the top, that L stands for larva. And what we find on mice is only the top 2 stages, larva, nymph, never adult, never. So only the juvenile stages suck blood from mice. And the thing I want to point out is, can you see them before I put the arrows? Those are them, right? These things that are about period size. That's what we're like hunting for on mice when we do this work. It's not the stuff you encounter. You have to go out and catch mice usually to find these things. Okay. All right, so this work took place at the WIU's field station. There are a series of long-term plots that Dr. Jenkins has set up to look at the effects of fire on a variety of things. Ebby's work covers 3 years where we collected animals. Her job was to identify ticks. Estimate abundance with the number of ticks per host, and then try to understand which variables determine tick infestation. In this Google map, WIU is here, Kibbe is west along the Mississippi River. In this blow up of Kibbe, this dotted red circle is our study area. On the right are these 15 plots, 2 transects each, where we would trap for 3 days, collect the mice, and dissect them. Oh, so again, so I've been doing this for 10 years. I think it's over. I think, right? 10 years is sort of a round number. I have gone out every summer, caught mice, looked for all their parasites, recorded it. And so I've had a variety of honor students working with me: Ashley Hitty, Janie Miller, Mary Street, Rachel and Quinn just helped me. Before I went to Poland, I came back from Poland and was desperate, and John Nichols and Sarah Obregon helped me. Sarah is a student studying raccoon genetics. Then is Andrew Hockenberry and Kofi Prempeh-Pujor. This is Fabian Payo, Sarah Kryling, and Kevin Delgado, Matthew Clayton, and that's Eddie. Matthew's actually doing tick work out east. He just emailed me today saying, we're doing this project where we're throwing fungal spores out in the environment to see if it kills ticks or not. So he's the guy in the purple cap. Oh, and there's the last batch. Okay, so here's Matthew again. Here's Joe Shesky and Sylvia Barclay, who was a student from someplace else, but her mom is the chair of English. So she worked for me this summer. Okay, to look for ticks, I've killed the mice, we brushed them off. We look for ticks. There's Ebby doing it. They're tiny, remember? So she's probably got a tick here that you can't see. We use these funky magnifying goggles. But here are ticks on a mouse here where they often are. These 2 are full of blood, but you have to use a microscope often. These 2 are attached but not full of blood, and they totally blend in. Okay. All right. So every year I would collect these mice, throw all the ticks in a vial, and start teaching genetics or something every year. So I have a decade's worth of ticks in tubes that need to be identified. So what Ebby did with David Hernandez is figure out how to identify ticks, and she found only 2. So the dog tick and the deer tick. This is actually, I think, an important result because Lone Star ticks are out there. I went to a CDC conference where they said, Lone Star ticks won't feed on mice. And at the end of every talk, I would say, did I hear you say? And they say, yeah, but no one has published it, like ever. I found one paper previous to this that where all three species live, you only find these two on Peromyscus. So I think that's very cool. All right, so the analysis was this. The outcome or responses were the abundance of these two species. We asked whether mouse attributes like sex and mass matter, whether the year we trap mice matters, also some random effects. And so these are the results. So the first question is, does sex affect infestation? And the answer is yes. And so there's a significant and consistent pattern where male mice have more ticks. So this x-axis is female versus male. The y-axis is the predicted abundance from the generalized linear model. Red is dog tick, blue is deer tick. So yeah, so significant effect of sex, significant effect of species. So dog tick numbers tend to be generally higher than the deer tick ones, and I'll say more why in a minute. And this sex effect, where males have more, is consistent. It's true for both species. And that's this table over here that says there's a non-significant species by sex interaction. So the effect of sex on tick abundance is the same for both. Males have more for both species. Then we looked at the effect of mouse size or mass, and here the two ticks respond differently. So there's a significant species by mass interaction, which means the effect of mass is different on deer ticks and dog ticks. And you can see the deer tick numbers increase with mouse size. The dog tick numbers do not. And I think all I can say at the moment is I haven't had time to start speculating why. And then the last thing I'll say is that what year we caught them in had a significant effect too. So there's a significant effective year. And I'm showing that in two ways. So these are the sex graphs and the mass graphs again, but split up by year. And you can see in the left-hand side, Dermacenter and Ixodes numbers are similar each year. But in 2023, the number of dog ticks explodes. So the sex difference is, what I think is cool is the sex difference is consistent year after year after year, but the total dog tick number blows up. This is the mass relationship on the right. And again, dog tick numbers go up, but every single year, the pattern is the same. Dog tick numbers are not affected by mass. Deer tick numbers are affected by mass. There was a significant random effect. So some plots in some years varied. I don't think I want to say more, because the last thing I'll say is like, after taking into account all those different factors, there was a correlation between dog tick and deer tick numbers on a mouse. So these are residuals plotting, meaning we have these counts of ticks after you sort of like factor out all the variation explained by sex and mass and plot, etc. There's still some variation that is a correlation between these two numbers. So we don't know what that is, right? Differences in immunology of hosts. And so we have more work to do. So my final thing here is that from this study, from Ebby's study, we have found that there are multiple drivers of infestation by ticks, and these two species are not always responding the same way. That, I think, has important implications for tick control, right? If all the predictors were the same for every species of tick, it would be much easier to figure out how to control them. But if every tick is responding to a different environmental or host attribute, then tick control becomes way harder. Okay. A new tangent that I'm not going to talk about is several students are now working on either bacteria in mice or protozoa in mice. So Kofi has done some blood microbiome work. Sada did blood and spleen microbiome. And Joe looks as though he's working right now on protozoa in mice blood. So my conclusion to this. So these parasites, they're everywhere. That's what ubiquitous means. They're really important for health or maybe population control in the wild. And in parasitology class, I say they're super freaking cool, right? They look awesome under the microscope, and talking about all of their life cycles and harm is really, really fun just with your friends and family, okay? I have shown you that there are a variety of things that drive infection levels. So if you're doing this kind of research, it's important to look at many. Yeah, I have to say, I feel really, really lucky that I've had this job for 20 years, being able to catch mice and do this stuff and learn things. I couldn't have asked for a better one. And With that, I say thank you and goodbye.
So, for those that can stick around, maybe we can ask a few questions and,
We can talk up to Dr. Tom.
what do you think about that? Okay. You guys, 10 minutes.
Oh, there, in the back.
So. Is it common for there to be two kinds of ticks on a mouse?
Oh, that's a good point, and one of the important things I didn't say. So, I haven't, so Abby and I have found almost no papers where this is described. So it could partly be because these ticks are thought to have different habitat requirements, so they may not often occur in the same place. But also, when people do tick research, they almost, you know how I was bitching about ticks this, ticks that? It's not only that, right? In the tick world, it's deer tick this, deer tick that, because they transmit Lyme disease. Deer ticks are all anyone cares about. So sometimes we found papers where the paper says there were dog ticks, but we ignore them for this, yeah, people just don't write about it. So I think it's another lucky thing that out at Kitty, all these are out there. So it's a really great opportunity for Evie and us to write about like these two ticks at one time. So, yeah, thanks.
I was curious if, what, do you think that a genetic bottleneck would, in a host species, would also affect the genetic diversity of the parasite species? And if so, how would you speculate that that would affect it?
Well, so I mean, if the parasites go through a bottleneck, then they're gonna lose diversity also. But now I have to think about, so like stuff I haven't read for years and years and years. But it might be that generally, and there are probably papers on this, but I don't know. There's less genetic diversity in parasite populations initially, right? Sometimes parasite populations are the organisms in a host. Those are often bottlenecked, right? You get one or two worms, that population size inside me is small. So it might be that parasite populations on average are less diverse than their hosts. And a bottleneck of the host that reduces their diversity will reduce the diversity of the parasites, but there's not much to start with, so it's not gonna change much. So that's me doing math on my feet, so, you know, different? Yeah.
Yeah, no, it's interesting.
Yeah. No, so it's really, really good, and so. Yeah, so I didn't say anything about like, all of the molecular details about genetic diversity in hosts and parasites and whether the diversity of the host affects who they can infect. Yeah, so it's really super interesting and cool stuff that I don't know about.
What's your favorite paper to write?
I think that first one, the island one, it's sort of small and simple, but I think it's one of the coolest things I've done and did. There was a review paper that came out like 2 years ago, and so I talked to Dr. McPherson about this, but in this paper, a review of genetic diversity in multiple populations, what do we know? That's the paper that said Meagher in one of the first, blah, 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 and they quote me. I mean, it's like, they actually quote words I wrote. And those of you who read, that doesn't happen very often, right? So they say something, and I said, and I thought, wow, yeah, I could have been a pretender. So it was just really fun, right? I think I wrote grant proposals, so thank you for asking this. I wrote a grant proposal that was totally slam, because they said, we know the answer. But I think it was a plant biologist where they'd done a lot of crop, monocrop, not monocrop. And so this reviewer said, you don't need to do it, we know. I gave a speech at a meeting once, and someone said, why didn't you look at like heterozygosity of individuals versus populations? I said, well, I've done some reading. I think, it doesn't matter the genetic mechanism for resistance, which I don't know in this case, but no matter the theory or the math, we expect this population level pattern. And this guy, the most famous genetic diversity and fitness guy in North America, in a meeting in front of a room of 40, said to me, you are sorely mistaken. And I was like. Actually, I was chairing the session, right? So I slithered to the side and said, our next speaker. So I think I was sticking my neck out. I thought I was a little crazy, but I really, really like it. I mean, it was like super fun. But so maybe for grad students, I'd say, if you find something you're interested in, you should do it, even when the big dogs are saying, you're really stupid, then you're. So, yeah, I think that's like, I've done a lot of stuff that's really fun. I've really enjoyed just seeing whatever pattern is out there. But, I was like a new grad student, just totally into figuring something out. Yeah, it was really my baby. So I like it, yeah. Okay, there's a class coming in, so that's the end. Thank you. Thanks, out there. Hey, Shawn. Hey, Ryan.
Hi.
I know you're not gonna retire.
Thank you, Shawn.
You're welcome. Happy to hear you.
Yeah.
I was gonna say, thing that got out, your registration is live tomorrow.
Oh.
So, what
I'll give you. I don't.
Thanks, Danny. So, Jim,
Yeah.
so we can still communicate, is that right? Like email?
Yeah. You know what? Let me send you an email with all my contact info. So, are you staying in Macomb?
We need to catch up.
Let me, 1 second. I never bought them. They brought some stuff.
Yeah, we need to catch up.
When's your last day?
This is last week of class, this next week is final, so like after finals. Good.
Yeah, let's have a Zoom meeting sometime.
Okay, and I know how to Zoom, right? COVID taught me that.
I'll send you a link.
Awesome to see you. Okay.
Okay.
Bye.
Thanks for the invite. Yeah, it's great.
I'll see you.
I want to stop recording.